So, welcome. If you're listening, you're probably trying to get your head around something complex, right?
Especially when it concerns, the well-being of people you care about, your approaches at parents.
Exactly. And you want the real core information, but, well, clearly and efficiently delivered.
Mm-hmm.
And that's what we aim for here. We dig into the research
Pull out the key bits.
and basically give you a direct path to understanding.
And today, we're really focusing on research that could be particularly relevant for, as you said, approaches at parents, the close relatives and parent-supporting individuals with 22q11.2 deletion syndrome.
Right.
We're looking at a really interesting article. It was published in Science Advances, March 12th, 2025.
Okay, let's flag that. The title is Synaptic-dependent developmental disconnectivity in 22q11.2 deletion syndrome.
That's the one. By Filomena Grazialvino and her colleagues.
Got it. So our mission today, let's unpack what this study tells us about how brain connections develop and change with this syndrome.
And importantly, what the potential implications might be for families, for those providing that day-to-day support and love.
Okay, so where did they start? It involved mice, I believe.
Yes, they used a mouse model for the syndrome called LgDel mice. And they found something pretty striking about how their brains were connected as they grew up.
It wasn't static.
Not at all. It was dynamic. So in the younger juvenile mice, they observed widespread hyperconnectivity.
Meaning too much connection, like
Right.
overly chatty brain regions.
You could think of it that way, yeah. An unusual amount of crosstalk between different areas. But here's the interesting part.
Okay,
As these mice went through puberty, that pattern actually flipped. It shifted towards a more localized hypoconnectivity.
less connection.
Less connection. Okay. Where specifically?
Primarily in the hippocampus,
Yeah.
which, as you know, is crucial for memory, learning, things like that.
Right. So a real developmental switch. Did they figure out why?
Well, they found a strong correlation. These connectivity changes in the mice seem to happen right alongside changes in the density of dendritic spines. Dendritic spines.
Mm-hmm.
Those tiny little projections on neurons where synapses form, the actual connection points.
Exactly. So it wasn't just the overall network map changing. It looked like the physical building blocks of the connections were changing too. It pointed towards a synaptic cause.
That makes sense. So the micro levels seem to match the macro level changes. Did they test that idea further in the mice?
They did. They used, a clever technique. They temporarily inhibited a specific enzyme involved in synaptic function. It's called GSK3.
Okay, GSK3 beta. What happened when they blocked it?
Well, temporarily blocking it actually normalized, or partly normalized, both the brain connectivity patterns and the dendritic spine density in the mice.
Wow. So that strongly suggests this GSK3 pathway is a key player, doesn't it?
It really does. It points to a specific molecular mechanism potentially driving these developmental shifts they were seeing in the mouse model.
Okay, that's fascinating in the mice. But the big question always is, does this translate to humans with 22q11.2 deletion syndrome?
That's the crucial next step they took. And the answer seems to be yes. Remarkably similar patterns emerged.
How did they show that?
They analyzed a large set of existing fMRI brain scan data from people with the syndrome, looking across different age groups.
And they saw the same switch.
Essentially, yes. A pattern of hyperconnectivity, sort of like the young mice during childhood, and then a transition to hypoconnectivity after puberty.
That's a really strong parallel. Amazing.
It is. It suggests this developmental trajectory might be a core feature.
Which parts of the brain were involved in humans? Was it the same as the mice?
There were some overlaps and some differences. The childhood hyperconnectivity in humans was mainly in posterior temporal areas.
Back and sides of the brain,
Yeah,
processing sensory info.
exactly. And also somatomotor regions, movement and sensation, and something called the dorsal attention network.
Okay. And the hypoconnectivity after puberty.
That was most prominent in the hippocampus, just like the mice, and also the temporal lobe more broadly.
Temporal lobe,
Yeah.
memory, language.
That's right. So again, key areas for cognition and processing.
So puberty really stands out as this critical window, a pivot point for brain connectivity development in the context of 22q11.2 deletion syndrome.
It certainly seems that way based on this research, a really important period of change.
And the underlying cause, based on the mouse work, points towards the synapses.
Yes, the evidence strongly suggests these large-scale network changes are rooted in what's happening at the synaptic level. The mouse work with the dendritic spines and GSK3 provided pretty direct evidence.
How did they bridge that synaptic link back to humans without, obviously, directly measuring spines?
They used a really smart technique called gene decoding analysis.
Okay, what does that involve?
They looked at the human brain regions that showed those developmental connectivity changes the hyper to hypo shift, and they asked, are these regions genetically different in some way?
And were they?
Yes, they found these specific regions were enriched, meaning they had a higher concentration, of genes known to be involved in synaptic signaling, how synapses function.
So the genetics of those changing brain areas matched the suspected biological function.
Precisely. And even more specific, these regions were enriched for synaptic genes that are known to interact with GSK3.
The same pathway from the mouse experiment.
Exactly. It really ties the human brain imaging findings back to the molecular mechanisms suggested by the mouse model. It's a powerful connection.
That is neat. Did they mention other genes? Because 22q11.2 involves the deletion of multiple genes, right?
They did. They acknowledged that other genes deleted in the syndrome they mentioned, COMT, DGCR8, RNT4R, SEPT5, likely contribute to, possibly through their own roles in synaptic function or interaction with pathways like GSK3.
So it's complex, but a picture emerges of disrupted synaptic development and function being key.
That seems to be the core message, yeah. With GSK3 potentially playing a significant, though maybe not exclusive, role.
Okay, let's shift gears slightly. Thinking about the approaches of parents listening, what does this mean clinically? How does it connect to what families might experience? The study mentioned links to neuropsychiatric conditions.
Right. That's a crucial aspect. We know that individuals with 22q11.2 deletion syndrome have a higher prevalence of conditions like autism spectrum disorder and schizophrenia.
Okay.
And what this study found was that the specific pattern of brain disconnectivity, they identified that developmental shift spatially overlapped with areas linked to gene expression changes seen in both schizophrenia and autism.
So the brain regions changing are also regions implicated in these conditions.
Yes, particularly for autism. They found a strong enrichment for genes that are typically less expressed or show reduced expression in individuals with autism.
That's a very direct link.
Yeah.
Does it connect to observable behaviors like social interaction?
It appears to. This is really relevant for approaches at parents. The researchers found a correlation in the human participants between the connectivity patterns in those sensory motor regions we mentioned earlier and their social responsiveness scores.
How so?
Well, greater challenges with social interaction, what they termed social impairment, were linked to the hypoconnectivity pattern in those regions during childhood,
Yeah.
and then linked to the hypoconnectivity pattern in those same regions in adulthood.
So the changing brain connectivity pattern tracked alongside differences in social characteristics across development.
It suggests a potential biological underpinning for some of the developmental changes in social abilities that families might observe. It's complex, but the correlation is there.
Did the mouse studies shed light on the behavior link? Did blocking GSK3 help with sociability in the mice?
That's where it gets more nuanced. The LgDel mice did show reduced sociability, as expected. But interestingly, the temporary GSK3 inhibition didn't fully rescue or reverse those social deficits.
Oh, so it fixed the brain connections and spines, but not the social behavior.
Not completely, it seems, which suggests that while GSK3 is involved in the synaptic and connectivity aspects, social behavior itself is likely influenced by other factors too, perhaps other genes in the deletion or different pathways.
But did the GSK3 inhibition help with anything else behaviorally in the mice?
Yes, it did partially rescue memory deficits in the adult mice.
Okay, so a more direct link there
Yeah.
between that pathway and certain cognitive functions, maybe more so than complex social behaviors.
That could be one interpretation, yes. It also highlights that the relationship between brain connectivity, even when altered, and specific behaviors isn't always one-to-one. The study itself mentioned that the typical brain behavior relationships seem disrupted in the LgDel mice.
It really underscores the complexity, doesn't it?
Absolutely. While we're finding these biological clues, translating them directly to behavior is still a major challenge. For approaches of parents, it reinforces that their loved one is a whole person, not just a set of brain scans.
Definitely. So let's try to synthesize this for the approaches of parents listening right now. What are the really key messages, the sensitivities to take away from this research?
Okay, well, I think first, it provides a deeper biological context for understanding brain development in 22q11.2 deletion syndrome. It shows it's a dynamic process.
Not a fixed state.
Exactly. Especially that period around puberty seems really important. Knowing this can maybe provide a helpful framework for families supporting their loved ones through different life stages.
Understanding that changes are happening biologically.
Right. And acknowledging that this manifests differently in everyone. The study points to mechanisms like synaptic function, GSK3, but it absolutely doesn't suggest a simple cause or a single outcome.
It's not a blueprint for every individual.
Not at all. It highlights the complexity, the multiple factors involved. It reinforces the uniqueness of each person's experience with the syndrome.
And what about the link to social characteristics? How should families think about that?
I think it offers potential insight. Seeing that brain connectivity patterns correlate with social development might, down the line, inform how we understand and support social skills.
Maybe timing interventions differently or focusing support based on developmental stage.
Potentially. But it's crucial to stress this is ongoing research. Our understanding is still evolving. For approaches at parents, it's another piece of the puzzle, offering a biological viewpoint that can sit alongside their own invaluable observations and experiences. It's not the whole picture, but it adds to it.
That's a great way to put it. So wrapping up our deep dive today,
Yeah.
the big takeaway seems to be this significant developmental shift in brain connectivity tied to 22q11.2 deletion syndrome.
Yes. Moving from that childhood hyperconnectivity to post-pubertal hypoconnectivity, likely driven by underlying synaptic changes.
With GSK3 emerging as a potentially important player in that mechanism.
And we see possible links, though complex ones, between these changing brain patterns and observed characteristics, particularly around social development.
Right. And just to remind everyone, the source for this was the Science Advances article.
Synaptic-dependent developmental dysconnectivity in 22q11.2 Deletion Syndrome by Filomena Grazia Alvino and colleagues. Definitely worth a look if you want the full details.
It really does show how valuable understanding the biology can be, even with complex developmental conditions. And research is clearly pushing forward.
Absolutely.
So maybe final thought for approaches at parents listening. Considering this dynamic picture of brain development, the shifting landscape, particularly around puberty, how might that inform your perspective, your understanding, and perhaps even the ways you support your child or loved one with 22q11.2 deletion syndrome as they navigate these different chapters of life? Something to reflect on.